Good day, this is Professor Wills, MD, PhD, and today's Friday, April 29th, 2022. It is 4:38 and 57 seconds, AM Eastern Standard Time. Today's research, breast cancer recurrence. So let's hop to it here. The new factors that can predict breast cancer recurrence. Defined. Discovery has the potential to also benefit treatment choices. Date April 22nd, 2022. Source, Georgetown University, Medical Center. Genetics and other factors that can determine if a woman is at risk for a reoccurrence of breast cancer have been identified, providing new research avenues for preventing a new tumor from developing. The researchers focused on breast epithelial cells, which are the layer of cells that form the ducts and lobes, which make milk during lactation. The investigators extracted these cells from donated non-cancerous tissue in the same breast as the one that had cancerous tissue removed during a mastectomy. The scientists were looking for numerous factors that can kickstart recurrence, but their main target was the entire collection of RNA sequences in the cell, the transcriptome that helps determine when and where each gene is turned on and off in the cell. Even though surgical techniques continue to improve, undetectable microscopic pieces of tumor can remain. There are one factor for reoccurrence of breast cancer in up to 15% of women. And sometimes years after surgery, people with hormone receptor positive breast cancer have the highest risk of recurrence. When analyzing expanded epithelial cells from women who had chemotherapy before their surgery, the researchers found significantly altered RNA. In particular, they saw significant changes in genes that had previously been recognized as prognostic indicators for cancer. And when a person is diagnosed with breast cancer, researchers have several tools, including testing for genes, such as BRCA1/2, to decide whether they should get certain kinds of chemotherapy or just receive hormonal therapy. But the tools we have available today are not as precise as we would like. About 1 in 8 women are diagnosed with breast cancer in the developed world. Researchers hope that their findings will help lead to more precise and directed screening in the future, and sparing women unneeded procedures, as we currently screen almost all women between the ages of 40 to 70, sometimes very aggressively. The researchers also noted that there are implications for women who have not had breast cancer, as some of the RNA alterations were linked to memory stem cell formation. The stem cells are self-renewing and related to growth and development. Memory stem cells are adult stem cells that can differentiate or change function into specialized memory epithelial cells. And if these cells get dysregulated, there is an increased potential for cancer. The cells from pregnant women were of particular interest to the researchers, as pregnancy usually triggers extra renewing cycles in the cell, potentially increasing the risk of cancer. And this study used conditionally reprogrammed cells for the initial isolation of epithelial cells. Conditionally reprogrammed cells is the only known system that can indefinitely grow healthy as well as cancer cells. Up to 1 million new cells can be grown in a week. Heretofore, one of the key problems in studying these cells was that epithelial cell cultures were often contaminated with other cell types, particularly fibroblasts, which grow very quickly in culture, while epithelial cells grow a bit slower. The primary tumor cells also can be difficult to isolate, but the researchers had increased success using the conditionally reprogrammed cells technique compared to conventional methods. Anything that researchers can do to prevent the occurrence or reoccurrence of cancer is a significant step forward. For researchers' sake, this may be an important contribution to reducing misdiagnosis, as well as a point, 2 ways, to develop better therapies to treat the disease. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.